Hello, my name is Kiran Batta. I'm a senior lecturer in the Division of Cancer Sciences at the University of Manchester. In my lab, we study epigenetic dysregulation leading to leukemias with a focus on a rare type of leukemia called chronic myelomonocytic leukemia, or CMML in short. CMML is a stem cell disease and it is characterized by monocytosis, that is, excessive number of monocytes. So in this lecture, I'm going to discuss the role of stem cells in leukemias. I'm going to cover the following topics. I'm going to start with hematopoietic stem cells, or HSCs. What are hematopoietic stem cells? What are the characteristic features of hematopoietic stem cells? How do you identify these stem cells? And how do you quantify them? And is there any heterogeneity in hematopoietic stem cells? If so, how do we evaluate that? And what is the functional relevance of such heterogeneity? Then I will introduce you to a concept called age-related clonal hematopoiesis ARCH, or CHIP, which is clonal hematopoiesis of indeterminate potential. Okay. So how clonal hematopoiesis presents a risk for hematological as well as non-hematological cancers, but also for other diseases like stroke, diabetes, and HIV. I will also discuss how CHIP influences drug response and predict relapse. Then I'll talk about leukemic stem cells, their cell of origin, how do you define them, their characteristic features, how do these leukemic stem cells respond to chemotherapy, how they are involved in a disease relapse. Finally, I'm going to talk about targeting these leukemic stem cells. As you probably know, leukemic patients also have got residual normal stem cells. So these are the stem cells that are not mutated, but somehow they're not doing their job properly. So I'm going to talk about how leukemic cells hijack normal hematopoiesis. Finally, I'm going to talk about advances in technologies that's helping us understand the pathogenesis of leukemia. That includes disease initiation, drug response, remission, and relapse. You're all probably familiar with this slide. Hematopoietic stem cells sit at the top of the hematopoietic hierarchy, which can divide and differentiate into restricted progenitor cells like common myeloid progenitor cells or common lymphoid progenitor cells. And these progenitor cells divide and differentiate into all types of blood cells you see down below here. Usually, this is a well-controlled process. As you may know, body makes about 2.5 million cells per second, and this is all driven by hematopoietic stem cells. So, what are hematopoietic stem cells? So, one can define hematopoietic stem cell as an immature cell that can develop into all types of blood cells, including white blood cells, red blood cells, and platelets. So by this definition, what we say, long-term hematopoietic stem cells, short-term hematopoietic stem cells, multipotent progenitor cells, all should be called hematopoietic stem cells. So a better definition is needed, which could be that hematopoietic stem cell is a rare type of cell present in blood or bone marrow that can self-renew as well as differentiate into all types of blood cells. The question is, how do you distinguish self-renewal from differentiation? Normally, hematopoietic stem cells are quiescent or dormant. They are in Z0 phase of the cell cycle. Okay? And then they turn to active HSCs. So these are the ones that can divide and differentiate and are usually in G1S phase of cell cycle. So the transition from dormant to active HSCs is reversible. Active HSCs undergo symmetric or asymmetric cell divisions. So HSC can divide and gives rise to two HSCs or two HPCs via symmetric division. HPCs are the progenitor cells that can divide and differentiate to all types of blood cells, but they can't self-renew. Via asymmetric division, It can both maintain the stem cell pool as well as differentiate to HPCs and then meet the demands of the body. So the first question is, what controls the transition from a dormant HSC to active HSCs? So Andreas Trump group in Heidelberg have looked into this. So they sorted for dormant HSCs by a marker called GPRC5C and performed single cell RNA sequencing. They found that the transition from dormant HSC to active HSC is a continuous process rather than you've got two distinct populations like DHSCs and AHSC. Okay? With the bulk cell data, we would not be able to draw these conclusions. They found a continuous increase of biosynthesis, increase in metabolism during transition from dormant to active HSCs. Dormant HSCs show high vitamin A induced retinoic acid signaling. The high levels of vitamin A prevents the activation of stem cells. Okay? And whereas vitamin A deficiency exhausts stem cell pool because it induces differentiation. Okay? So what else regulates the transition from dormant to active HSCs? For example, cell surface markers like CD38, while regulating the release of calcium, can regulate dormancy. And of course, the bone marrow niche where the hematopoietic stem cells are residing, that can also regulate dormancy. So the next question is, what regulates symmetric versus asymmetric division? Stress or inflammation promotes symmetric divisions to HPCs to restore hematopoiesis. Aging also contributes to symmetric division, and oncogenic mutations like DNMT3A also cause symmetric divisions. For asymmetric divisions, cell polarity is one of the major determinants. Cell polarity is often lost in leukemias, okay, and that contributes to asymmetric divisions. Okay? Some fate determinants like RNA binding proteins also contribute to asymmetric divisions, and asymmetric inheritance of mitochondria or epigenetic marks like HCTK27 trimethylation during cell divisions can contribute to asymmetric cell divisions. So the next question is, how do you identify HSCs? So traditionally, HSCs are identified by their ability to reconstitute or replenish entire blood system of an immunocompromised recipients in a process called transplantation. Then you can't really study those cells because you've already transplanted them. Is there any easy way to identify HSCs? The short answer is no. For decades, scientists have been trying to identify cell surface markers that could define HSCs. In humans, if a cell expresses CD34 and CD90 and CD49F and lacks any lineage markers or CD38, then they can be called as stem cells. But again, if you sort these cells based on these markers, not all of them will be HSCs. You can only enrich for HSCs, but they're not all HSCs. Okay. And recently, people have come up with additional markers like CD273, which further enhances the enrichment of HSCs, sometimes up to 1 in 2. But again, it's not a definitive proof that they're all HSCs. How do you know that? There is an assay to quantify HSCs. It's called limited dilution assays. As I said, if a cell is a true HSC, you can take a single cell, transplant into a conditioned recipient, and of course, it's difficult to transplant single cells into mice. So how do we quantify the number of HSCs? So what we do is we transplant different numbers of cells and score for the mice, okay, that did not show any engraftment. Using this method, in this graph, you can see we can quantify the number of HSCs. So roughly around 1 in 12 cells is a HSC. But this is a difficult experiment involving many mice. So recently, well, not recently, more than a decade or so, Liu et al. Developed a new method called barcoding technology. So the lentiviruses are used to deliver DNA barcodes in, into cells, such that each cell receives only one barcode. You can transplant these cells into mice. Okay. And after 16 weeks, you isolate distinct populations of cells and sequence for the barcodes. Okay. If you see a barcode in HSC, then you see the same barcode in all mature cell lineages. Okay. Then you know that HSC contributed to multilineage contribution. Based on this data and recent single cell transcriptome data, models of hematopoietic differentiation has been changed. So this is a classical model of hematopoietic differentiation, which suggests that lineage commitment, okay, is associated with branching transition between the cells with distinct lineage potentials. Okay. So the new data suggests that HSC differentiation occurs via a continuum, okay, and there is more plasticity than previously. So recent work from last Mantle group performed clonal tracing, basically the same set of experiments, not using lentiviruses, but using naturally occurring CpG methylation marks as barcodes. Okay, so there's no genetic manipulation or lentiviral transduction. So using this method, they found that during aging, a small number of expanded clones contribute to myeloid bias, but you also have somewhat younger clones with multilineage potential. This again suggests that hematopoiesis is polyclonal, still polyclonal even at old age.